Good morning, everyone. It's my pleasure to be the moderator for this session. So the first plenary session for today is the Professor David Cooper Memorial Lecture. And I'm sure you all know David very, very well. David was an eminent Australian immunologist, the founding director of the National Centre in HIV Epidemiology and Clinical Research, now the Kirby Institute at the University of New South Wales, Australia. He was also one of the founding co-directors of HIV-NAT. David's life was dedicated to the prevention and treatment of HIV and other infectious diseases, and he contributed to the development of almost every drug used in HIV treatment to date. I cannot think of a better person to give this memorial lecture than Professor Tony Kelleher. Tony is an excellent clinical immunologist and laboratory scientist. He is the current director of the Kirby Institute. Tony has an outstanding reputation in the field of immunology and HIV research and played a key role in multiple clinical trials. His research spans from the basic science of HIV pathogenesis to HIV preventive vaccine development to HIV treatment, and more recently to the block and lock strategy for HIV remission. The title of his talk is Future Directions for HIV Therapies. Thank you, Tony.
And it is a great honour, as always, to be at this symposium, which I think is an incredible initiative that's now run for the last 22 years. And it's an incredible honour to be giving this David Cooper Memorial Lecture. And I think it's somewhat humbling to be introduced by by one of the people who I think is one of the emerging leaders in the field. So David, as Denise has just said, was an inspirational leader. He was a mentor and a friend. He was first and foremost a clinician scientist. He believed in data, not expert opinion, and he spent his research career ensuring that data was gathered in a high-quality way. And that really has underpinned much of the strategic use of antiretroviral therapy for the treatment of AIDS that is still used today. He believed in health as a human right and believed in treatment access for all, and he pursued this vigorously in many guises, but in particular when he was president of the International AIDS Society. And he was committed to this region and particularly to capacity building. And he was incredible at identification and support of talent and growing that talent to ensure the next generation. And there is a picture of the three amigos who really underpin the fact that we're here for the 22nd time. So these are my conflicts of interest. So, as I've just said, antiretroviral drugs have revolutionized the treatment of HIV. Those drugs hold back the virus, they hold it in check, and they allow immune reconstitution if you start antiretroviral therapy late, and immune preservation if you start it early. And what we've learned over the last 5 to 10 years is that these same therapeutics are very, very good at preventing HIV in the form of PrEP. But compliance is an issue. So what does the future hold? Well, there will be further advances in these antiretroviral therapies. New classes are coming online, and there are currently many trials looking at long-acting antiretroviral therapy, both for treatment and for PrEP. And these will be dealt with by eminent speakers later in the program. These advances will be important for both compliance and choice. And the other major advances that need to occur in the future are those of access to therapy. And they also will be dealt with extensively in the program. So I've chosen to look a little bit beyond the immediate event horizon and try and give you some background on where the laboratory is meeting the clinic in a translational fashion, to try and look at ways where we might advance both the cure and remission strategies and the control of residual morbidities that still afflict patients infected with HIV. And in this talk, I'm not going to deal with immune activation, but I'm going to talk about the rise of immunotherapy for malignancy, which we've seen outside the HIV field, and how this might impact on the residual morbidities we see in virally driven cancers. So to deal with the reservoir first, HIV in the reservoir is in what we call a latent state. And most of this virus is in a form called post-integration latency. What does that mean? It means that the virus is in its DNA form, integrated into the human genome. It's there, but it's silent. It's not transcribing, it's not making RNA. And importantly, the key thing to understand is that that gene expression from HIV, from the genes of HIV, is driven much like any other gene. It's driven by the activity of promoters that sit in front of those genes. And in HIV, this promoter is referred to as the 5' LTR, or long terminal repeat. What are the mechanisms of latency? There is this thing called epigenetic silencing. So all genes are associated with histone structures within the genome that are based on histone architecture, such as nucleosomes. And the HIV LTR is no different, and it has a very particular histone architecture associated with that I will go into in some more detail in a moment. But the transcription of HIV is controlled by the deacetylation and methylation of those histones, which change the biochemical properties of those histones and their architecture. And epigenetic silencing also arises from sequestration of host transcription factors and viral factors such as TAT that regulate viral transcription. A latent virus may be latent because it's integrated into a silent gene. And you say, well, how does that happen? HIV preferentially integrates into active genes, but those genes may be active in a particular cell, and as that cell goes into a resting state or differentiates, those genes become resting. And so as those genes become resting, the virus also goes into a resting or latent state. Or the virus may appear to be latent and unable to transcribe, but it's in fact defective because it has a stop codon or it has an insertion or a deletion that makes it replication incompetent and unable to transcribe. So this diagram looks very scary, but it basically shows the whole genome of HIV with the 5' LTR expanded here. And these grey and pink structures shown here are the histones formed into nucleosomes associated with the 5' LTR. And there are always two of these. There's nuke 0 and nuke 1, not very imaginative names. And importantly, nuke 1 sits over the transcription start site. And in fact, it's well demonstrated in this retrovirus and also in HTLV1 that particularly changes in this, the architecture of this histone, this nucleosome, changes the orientation of this nucleosome, and actually it slides up and down the 5' LTR, revealing the transcription start site and allowing the virus to transcribe. The 5' LTR also contains all these host transcription factor binding sites that regulate HIV transcription. And as you can see, there's multiple different ones, each shown here as a different color. The important thing, the basic way to think about this is if you get methylation of these histones, you get a closed chromatin architecture and the virus is unlikely to be transcribing. If you get acetylation of these histones, you get an open chromatin structure, you get sliding of this nucleosome to reveal the transcription start site, and you get active transcription of the virus. And this biology has underpinned the shock and kill approach, which we've heard about at this symposium and elsewhere, particularly from lectures given at plenary lectures given at previous symposiums by people like Sharon Lewin. And Sharon and others have pursued this shock and kill approach, which is really based on giving various small molecule inhibitors that change the profile of those histones from one that's dominated by methylation to one that's dominated by acetylation to try and encourage transcription of the virus, have the virus produce its proteins, and then those cells that are producing active virus be killed either directly by the immune system or by things like neutralizing antibodies that are given at the same time. A less well-studied approach is something that we have been pursuing for quite some time called block and lock. And the idea here is really to do exactly the opposite, is the simplest way to think about it. So you can use various things like sRNAs and short hairpin RNAs, which I'll concentrate in this talk, but other small molecule inhibitors and TAT mimics to actually shut down viral transcription to hopefully induce something that we've called super latency that is resistant to host factor activation and lock the virus down into a silent state that is maintained for long periods of time. So this is what we're trying to do with our shRNAs. So we found, to our surprise, some time ago that if you take shRNAs that are aimed at the promoter of the virus, the 5' LTR, that match sequences with certain sequences within that promoter, you can change this open chromatin structure to this closed chromatin structure in the virus, and you can shut transcription down. So we have identified two lead candidates, which we call PROM-A and SH143, which bind to this area here, which happens to be the same area as the important transcription factor NF-kappa B binds. And we've found another one that's associated with nuke 0 that binds to near to the AP1 binding site, which is another important host transcription factor that activates HIV transcription. Importantly, these sequences are relatively highly conserved across various subtypes of the virus and do not have similar sequences within the host genome. So we try to maximize on-target effects and minimize off-target effects. We've got two of these lead candidates, but we've done a screen of this whole 5' LTR, and we have about 10 of these that look promising. And we found that doing some mathematical modeling that if we have 4 of these in combination, we can cover in excess of 99% of the variants that are described in the Los Alamos database. So what happens when you treat a culture with these? Well, the red and the green line here shows the effects of these two sRNAs, and these are various controls up here, which I'm not going to go through. But basically, without these, you get lots of virus. With these, you get very little virus. This is shown by P20 or by RT, reverse transcriptase here. But if we measure this by RNA, we get a 3 to 4 log drop in virus with one of these. And it lasts for one treatment with an sRNA in vitro lasts between 16 and 21 days. So we get profound and prolonged silencing of the virus, which is specific with minimal off-target effects. And we found that we can induce this in multiple T-cell lines, in monocyte-derived macrophages, and in astrocytes. Importantly, if you do the same sorts of cultures, but try and activate the virus with things like vorinostat, which is one of these HDAC inhibitors that has been used in the shock and kill approach, or TNF, which is a tumor necrosis factor which activates the virus. We can maintain silencing of the virus with either of our sRNAs. This is 143, this is PROM-A, and this is the combination. even at very high concentrations of these, well above what is being used in the clinic for reactivation, and well above what we see, the levels of TNF that we see in septic shock. So that's fine, that's all in tissue culture and petri dishes. We want to demonstrate this in vivo, and we decided to do this in SCID-HU mice, because in SCID-HU mice, these mice are reconstituted with a human immune system, and you can use HIV, whereas going into macaques, we would have to use different sequences and different delivery constructs to get it effectively into the macaques. So we reconstitute these SCID mice, these immunodeficient mice, with a human immune system, either using human CD4 cells or human CD34 stem cells. We allow those to engraft, and then we challenge the mice with virus. And so this is some of the results of those experiments, and you can show, this is again a measure of virus, the amount of P24 the virus is producing, and you get significant reductions in this very acute model of HIV infection. These mice without treatment die within about 4 weeks in this model, so it's very aggressive HIV infection. You get reductions in tissues of the amount of virus, and you get maintenance of CD4 T cells at the same level as mice that are uninfected. So, taking a lesson from what we know about antiretroviral therapy, we have pursued a combination approach in collaboration with an industry partner called Kelleher, who's recently been associated with CSL in Australia, who have an siRNA that knocks down CCR5, the co-receptor for HIV. And we've combined the delivery of this short hairpin RNA that knocks down CCR5 and prevents viral entry that way, with our shRNAs that induce silencing. So we're aiming at 2 stages of the virus life cycle. And this means that you get both prevention of viral entry, any virus that gets in gets silenced, and any virus that's there before you've gone in with your gene therapy that prevents viral entry will also be silenced by our siRNAs. So we think this makes biological sense. So we've set up a SCID-HU mouse model with our colleagues at the Walter and Eliza Hall in Melbourne, where we transplant a different group of a different strain of SCID-HU mice with CD34 cells that are transduced with various constructs. They're allowed to engraft. We then infect the mice with HIV, allow that infection to establish for about 3 weeks. We then treat them with antiretroviral therapy for a period, and then we take them off therapy and we look at what happens to the viral load. And so this is some preliminary results from these experiments. And you can see that this is now measuring the CD4 count in these mice throughout each of those stages. And you can see that in the 2 control groups, there is a decrease in CD4 T cells over time in the groups of mice. Whereas in the mice receiving the combined therapy, there is maintenance of CD4 T cells. There is also delay, though it's only minimal delay, in the groups treated in terms of rebound of viral load. And the numbers are still small there and preliminary, but we find this encouraging. The problem we have is that we need to get these therapeutics that work into more CD4 T cells. Currently, we use a retroviral approach that transduces about 40 to 70% of the stem cells. We need to increase that, and we need to get better entry into CD4 T cells. The current standard retroviral therapy approach is to use what we call VSVG pseudotyped retroviruses. And that makes sense for a lot of gene therapy, but it doesn't make sense for HIV. The receptor for VSVG does not exist on resting CD4 T cells. You have to activate the cells to get the virus in. And we don't want to reactivate the virus here. So we've pursued various ways of trying to develop new gene therapies in parallel with the new gene delivery vehicles. And we've taken two approaches. We're trying to develop new retroviral constructs, and I'm going to show you some data from that now, where we've reasoned that envelope from HIV gets virus into cells with these cells very, very well, so why not use envelope? And we've screened a large number of envelopes. We found one that is dual tropic and has low CD4 dependence, and this gets into very significant numbers of resting CD4 T cells of all flavors, whether they be naive, effector, central memory, slightly less indeterminately differentiated. We also want to, once the virus is in, we want to get more of it integrated, and VPX is a gene in HIV2 and in SOV that increases the rate of transport into the nucleus and integration in resting cells, and particularly in monocytes and macrophages. And you can see that here that VPX increases the rate of transduction of our constructs very, very considerably. And we've screened a library of VPXs, and we found one that equally supports transduction of resting T cells and monocytes and macrophages, which are the main homes of the reservoir. And so we are now building a retroviral construct where these two proteins are expressed in trans or into non-integrative constructs to see if we can do better with our transduction efficiency using this retroviral approach. With our colleagues at Melbourne University, Frank Caruso and others, we are all pursuing a different line of delivery where we're using various sorts of nanoparticles. One of those is shown here, a layer-by-layer nanoparticle, which we can load up with our siRNAs. And these are some fluorescent pictures of cells where blue is the nucleus, yellow is the virus, and red is our siRNAs when they've been delivered by these nanoparticles. And just to show you that these are in fact inside the nucleus. So this lump here is our siRNA embedded in the nucleus of a resting primary CD4 T cell. And without targeting these at all, we're getting about 15% entry into the nucleus and suppression of the virus. So we're now trying to target those more efficiently with antibodies and other ligands that will target resting CD4 T cells. So, to finish this section of the talk, I think we've got a suite of shRNAs that target the promoter of HIV that cause profound and prolonged silencing in multiple cell types of the virus. And the latency that this induces is resistant to T cell activation. And so I'd put it to you that at least in vitro we can induce this phenomenon called super latency. We have encouraging data in at least 2 separate murine models now, but our challenge is really delivery. The delivery of this to resting CD4 T cells is highly challenging, and we're investigating building better gene delivery systems, both by engineered retroviruses and layer-by-layer nanoparticles. I wanted to change gear now and talk about HPV infection and anal cancer. So as we know, HPV causes cervical cancer and other cancers, and anal cancer is analogous in many ways to cervical cancer. Infection of HPV causes increasing dysplasia of the epithelial cells in the anal canal, which becomes increasingly dysplastic and eventually transforms into a cancer. there is very high prevalence of the high-grade forms of this intraepithelial neoplasia in gay men, and it's even higher in men that are HIV positive. And this is an increasing problem and these infections are prevalent throughout the world, and particularly in this region. And the rate of these infections and the rate of transformation into cancer is higher in those who are HIV positive. So this is a graph showing the prevalence of HPV infection in gay and bisexual men. And you can see that the high-risk subtypes are prevalent in both HIV negative and HIV positive men, and often more prevalent in the HIV positive individuals at very high rates. Those data came from a study that was done in Australia called SPANK, where we followed 600 gay and bisexual men with anoscopy over a 3-year period to look at the progression of disease. I want to describe here a sub-study that we did about the correlates of progression and regression of high-grade disease in this cohort. So we chose to look at the immune responses in the periphery to the two oncoproteins associated with HPV that are called E6 and E7. And we did a range of T-cell assays, including intracellular cytokine assays and our OX40 assay, where we look for the upregulation of two activation markers, CD134 and CD25, to look at CD4 T-cell responses. And there's a lot of information on this graph, but if you look at the bottom corner here, what we found was that CD4 responses to E6, or the presence of those, correlated with regression of disease. That was published some time ago, but it got us thinking about how we might manipulate this natural immune response to encourage regression of disease. And so we took the opportunity of the fact that SPANK was finishing its follow-up, and our access to a drug called pomalidomide, which Mark Polizzotto at the Kirby had previously worked on at NCI with Bob Yakuin, and had demonstrated that pomalidomide could cause regression of treatment-resistant Kaposi's sarcoma, which we know is a virally driven cancer. So we designed this single-arm study called SPACE, where we gave pomalidomide to gay and bisexual men, whether they were HIV positive or negative, who had established high-grade intraepithelial disease that was not regressing. So they were given 21 days of pomalidomide out of each 28-day cycle, and they received 6 of those cycles. So there was 6 months of therapy, 3 weeks on, 1 week off, and they got 2 milligrams of pomalidomide. Pomalidomide is a sister drug to the drug thalidomide, and is currently licensed for the treatment of myeloma and myelofibrosis. But in myeloma, it regulates B-cell differentiation, but it is known to have significant impacts on the T-cell arm of the immune system. And so we argued that this is an orally available drug, relatively well tolerated, and we chose to look at its effect on the histological clearance of high-grade disease at anoscopy at 6 months, and then repeat that at 12 months when the patients had been off therapy for another 6 months. Therapy for this condition at the moment is mostly local and not very effective. And so although you can burn or freeze these lesions off, they tend to recur very quickly. And so really that 12-month time point was to look at whether we were getting sustained responses or not of therapy. So this is the study group. It was 26 men. They were aged about 50. They all had high-grade disease, and they'd all had that established for at least 3 years, so an average of 37 months. In the they were divided between HIV positive and HIV negative, and in those that were HIV positive, they had 700 CD4 T cells and were all virally suppressed. These are the results which surprised us. We'd powered this to detect about a 15 to 20% regression rate. We got regression rates of 52%, which were at least as good in the HIV positive men as the HIV negative men at 6 months. And this is the breakdown of complete and partial. And we got that sustained at 12 months. In fact, some of those that still had disease at 6 months converted to regression at 12 months. So there was an ongoing response after the withdrawal of the drug. And these are the waterfall plots of those 2 points shown here. So these are pretty impressive results. So along with that, we did a range of laboratory assays and showed that there was this really marked immune activation in both the CD4 and CD8 T cell compartments, which Mark and Irene Soretti had seen in the Kaposi sarcoma trial. So we replicated that. What they hadn't done was they hadn't done any functional studies, and we show that we get these really marked increases in the function of CD4 T cells, particularly in response to E6, but also in response to other viral antigens such as CMV. So we get these marked increases in both the response, but also in the background responses. So this is really inducing a lot of immune activation without any severe side effects. And it causes changes in CD4 and CD8 count and in the subsets, but all those changes are transient, and importantly, it doesn't drive out a senescent immune system. So we find this to be very encouraging data. We have used pomalidomide, which is a well-tolerated, orally available immunotherapy, and has shown now that this causes regression of 2 virally driven cancers, high-grade anal interepithelial neoplasia and Kaposi sarcoma. And this is associated with transient changes in the immune system, generalized immune activation, and functional changes in the CD4 T cell responses that we had previously shown correlated with natural regression of the disease. So we're still in the process of characterizing the responses within the epithelium and subepithelial, and still trying to understand how this actually drives regression of the disease. But I think the discovery of these immunotherapies that are much better tolerated and appear to be safe in HIV patients is encouraging for the future treatment of this form of morbidity that is very prevalent, particularly in this region. So I'll finish there by putting up my acknowledgement slide, because I did none of this work. All these people here at the Kirby and at our various collaborators did all the work. Thank you very much.